Welcome everyone. Today we are going to learn about membrane proteins. At first I will talk about two important facts about these proteins, then I will go to the main topic. The first point is that all cells contain plasma membrane and the basic architecture of the plasma membrane is lipid bilayer. But cells differ in their functions by the presence of different proteins in the plasma membrane. Like liver cells has plasma membrane and simultaneously the pancreatic cells and other cells contains plasma membrane. Both the plasma membrane in liver cell and pancreatic cells are lipid bilayer, but their protein compositions actually differs, and that's why their function also differs. So proteins determine the specific functions of the cells. Not only that, protein also specify the functions of intracellular organelles also, like nucleus, endoplasmic reticulum, Golgi complex, then lysosome, endosome, peroxisomes, mitochondria, chloroplast. All these intracellular organelles in the eukaryotic cells are also surrounded by membranes, and that membranes are also lipid bilayer, but the composition of the proteins in different organs are different. So protein composition differs in different cells and organelles. This is the first point. Next point is not only the protein composition differs, but the percentage of the proteins also differs. Like in case of inner mitochondrial membrane, the percentage of proteins is approximately 76% of the of protein, and only 24% is actually lipids. In general, cells contain 50% lipids and 50% cells, but sometimes the percentage differs. Like in case of myelin sheath that actually insulate the nerves, they contain only 18% of proteins. See how much difference is there. So not only protein composition, but the percentage of proteins also differs in different organelles and cells because of the purpose of the organelles and the cells. Now this is the first point. Then second point is there is three categories of proteins present. One is integral membrane proteins, integral, or also called transmembrane protein. Then this is the first category. Second category is lipid anchored protein, and third is peripheral protein. So at first I will talk about in this video the integral membrane protein in more details. The first point we have to remember about integral membrane protein. So I am removing these two proteins, lipid anchored and peripheral proteins. I will make separate video on these two proteins. First is integral membrane proteins. There is three important points we have to remember at first. That is, integral membrane proteins are amphipathic in nature. Amphipathic in nature. What is amphipathic? Amphipathic means those biochemical structure contain both the hydrophilic, that means water loving, and hydrophobic, water fearing domain. So if any biochemical structure contain both the hydrophobic and hydrophilic portion, we call amphi. Amphi means both philic. This is the first point we have to remember about the integral membrane. All integral membrane proteins are amphipathic. Both have hydrophilic and hydrophobic portions. Next point we have to remember about integral membrane proteins that they span the membrane. They traverse the lipid bilayer. Okay, that's why it calls transmembrane. And the third main point is they have three domains. Okay, now let me draw the structure of integral membrane protein. Let's suppose this is two lipid bilayer. This is the exterior of the cell, outside of the cell, and if this is the cytosol, then I can draw an integral membrane proteins like this. It has one domain. Let's suppose this is the second domain and this is the third domain. So this domain is called exoplasmic domain. Exo means outer side, plasmic domain. This domain is called that traverse. Basically, this domain actually traverses the lipid bilayer, so it's called transmembrane domain. Transmembrane domain. And this portion that present in the cytosol is called cytosolic domain. Cytosolic domain. Now, in case of integral membrane protein, this exoplasmic domain is actually hydrophilic in nature. Hydrophilic. This transmembrane domain is actually hydrophobic in nature, and the cytosolic domain is hydrophilic in nature. Hydrophilic. So both this inside and outside domain are water loving in nature, but the inside or the transmembrane domain is actually hydrophobic, water fearing in nature. Now, this portion is actually very important for this integral membrane protein. So I will talk about more details about this domain. Okay. The first point we can remember about this domain, transmembrane domain. Transmembrane domain, because this domain actually anchor this protein. Okay. So this segment traverse or span the membrane. So this point is actually covered by this transmembrane domain. So transmembrane domain is important, so we will talk about transmembrane domain. First is transmembrane domain of this protein is predominantly, it is found that this is predominantly alpha helix in nature. So protein has secondary structure. One type of secondary structure is alpha helix in case of protein. So predominantly this segment is alpha helix, but sometimes it also can be beta strands. So this is another secondary structure. Okay, multiple beta strands can be present, multiple, and also multiple alpha helix can be present. Okay, so the transmembrane segment can be alpha helix or beta strands. This is the first point. Next is that, because I have mentioned this is predominantly alpha helix, so I'm talking about at first alpha helix in more details. This domain of these proteins basically that span the membrane contains about 20 to 25 amino acids that are hydrophobic in nature, hydrophobic, and also uncharged. They must be uncharged. Now that's why, because this segment contains hydrophobic amino acids, so they are hydrophobic in nature. Now this portion of the lipid membrane basically has the fatty acids. So these all have fatty acids, and here the head group is present, that is polar in nature. So this is the hydrophobic core of the membrane. What we call this area hydrophobic core. Hydrophobic core of membrane. Now this segment is actually spanned by 20 to 25 amino acids that are hydrophobic in nature and uncharged. And the length these amino acids can make is approximately the length is 3.75 nanometer. That is quite enough to span the membrane. So this length is enough to span the membrane. This is another fact. Fourth point you can remember, this hydrophobic amino acids actually protrude its side chains. So they protrude, these hydrophobic amino acids protrude means they extend their side chains and makes van der Waals interactions with these fatty acids. Okay, now but they can interact with the fatty acids, but there is a problem. The basic bond that is present in a protein is called peptide bond. Let me explain this. So what I have told you that this portion is hydrophobic in nature. These segments contain hydrophobic, so there is no problem. They make van der Waals interactions. But the problem is basic in structure, the presence of peptide bonds. Remember this. As because this is a protein, sorry, cytosol. As because this is a protein, so protein must have peptide bonds. Now, peptide bonds is also present in this segment because there is 20 to 25 amino acids. So there must be many peptide bonds, and peptide bonds is basically polar in nature. Polar in nature, that means hydrophilic in nature, and this portion is actually hydrophobic in nature, so that will create problem. That's why it has been found that the peptide bonds is C double bond O NH. Okay, so the peptide bond, this bond of amino acids actually make hydrogen bond with another peptide bonds to seal the peptide nature of carbonyl group. Okay, polar nature of the carbonyl group. Now carbonyl oxygen, this oxygen makes contact or make hydrogen bond with 4 residue. You have to remember this, with 4 residue amino acids towards the C terminus. Now let me explain. Towards the C terminus, how it will look. Let's suppose this segment contains 20 amino acid segments. So let's suppose this is the first amino acid, then it has peptide bond, then a second amino acid, then a peptide bond, then third amino acid, and up to, let's suppose 20, oh sorry, at first 4. I want to make 4 amino acids, 4, 5, I mean, then up to 20 amino acids. Let's suppose this is the segment of transmembrane domain. So this is the transmembrane domain. Transmembrane domain amino acids. Okay. So these amino acids are hydrophobic in nature. There is no problem. But the peptide bond of every amino acid that make are actually polar in nature, because this carbonyl bond, oxygen can make hydrogen bond. So to prevent this polarity, it makes hydrogen bond with the 4 residue amino acid. That means if this is the first peptide bond, then this is the first peptide bond, then this is the second, this is the third, this is the fourth. So this peptide bond, this carbonyl group will make a hydrogen bond with this amide bond. That means if there is C double bond ONH group, that is the peptide bond, then this carbonyl group will make bond with the C double bond NH with this hydrogen. So this carbonyl oxygen will make a hydrogen bond with this amide hydrogen. So 4 residue apart amino acid towards the C terminus, the carbonyl group of the amide peptide bond will make hydrogen bond with the NH, that means this amide bond. So apart from 4 peptide bonds, it will continue to make peptide hydrogen bonds to seal the polarity. So what will happen after the first bond? Let's suppose from here bond is making here, with the second peptide bond it will make the here another hydrogen bond. With this it will make another hydrogen bond. So every CO carbonyl group oxygen will make a hydrogen bond with the NH, but apart towards the C terminus, because we know in case of protein the here the N terminus and here will be the C terminus. CWH group is present. So 4 amino acid residue apart from the amino acid towards the C terminus, it will continue to make hydrogen bonds. Basically helix look like this. So if this is the first first peptide bond is the 5th peptide bond, then it will make a hydrogen bond. Then second will make with the 6th hydrogen bond. That's why it shields the polarity of the peptide bonds. So this is another important point we have to remember. Now let's take some examples of integral membrane proteins. Let's suppose this is the lipid bilayer. One example of integral membrane protein is glycophodin A. Glycophodin glycophorin. This protein is abundantly present in the red blood cell in plasma membrane. And this protein, integral protein, has a single alpha helix. And I want to mention here that a single alpha helix is enough to span the bilayer. So these proteins look like this. So it has a single alpha helix and a cytosolic domain. So exoplasmic domain, transmembrane domain, and cytosolic domain. And this has only a single alpha helix. So it is called single pass membrane protein. Single pass membrane protein. So it has only one alpha helix that can actually span the membrane. But in case of glycophorin, coil structure can form. So two glycophorin comes close together and they actually make coil structure to make the dimer. And the dimer is actually functional basically. So one glycophorin and second glycophorin comes close and this transmembrane domain makes coil structure. So it is a functional protein. The dimer is a functional protein. Another example, let's take another example like aquaporin. This is actually a water channel protein. Also glycerol can pass through aquaporin. So this is an example of water channel proteins. Okay. Now aquaporin is actually tetramer in nature. Tetramer. That means there is 4 subunit. But each subunit, remember, each subunit contains 6 alpha helix. Each subunit contains 6 alpha helix. So there will be 6 times it will pass. So let's suppose this is the first time, second time, fourth, fifth, just like this. So there is multiple alpha helix present in the aquaporin and the number is 6. So that's why it is called multipass protein. So it is single pass, they are called multipass. Multipass means multiple times it spans the membrane. Another kinds of proteins are called G protein couple receptor. Another example is bacteriorhodopsin. This protein is very important and I will make a separate video on bacteriorhodopsin. This is an integral membrane protein. Also G protein couple receptor is an integral membrane protein. This is a group of proteins. Now these proteins actually span the membrane 7 times. So there is 7 alpha helix present. So let's suppose this is the first, second, third, fourth. So 1, 2, 3, 4, 5, 6 and 7. So look like this. So they traverse the membrane 7 times. So they have 7 alpha helix and also they are multipass under the multipass membrane protein. Now let's talk about some proteins that has beta strands. One example is show here. One example of beta strand proteins is called porin. Now porin is different from aquaporin. Remember, porin is different from aquaporin because aquaporin contains alpha helix and porin contains beta strands, multiple beta strands. Just like aquaporin is tetramer, this porin is actually trimer. That means there is 3 subunits present in the functional protein, and each subunit, remember, each subunit contains 16 beta strands that make a structure. Remember the name, that is called beta barrel. Okay, so this porins actually form a pore-like structure. Let's suppose this is the plasma membrane. Here the porin makes a pore-like structure. I'm just giving you the just a brief idea. This is not the perfect structure. There is strands of beta strands present that makes the beta barrel, but actually make it pore-like structure. So it's called porin, and small water-soluble molecules are actually permeable through the porin structure. Now, this beta valence structure are more abundant in the outer membrane of bacteria, mitochondria, and chloroplast.